The cell membrane is a physical boundary between the organized inside of the cell and its external environment. The cell membrane is semi-permeable and allows for a dynamic exchange and interplay between the cell and its environment. This video will examine the varying models that have led to the fluid mosaic model and analyze their limitations, whilst evaluating the cellular processes of passive diffusion, active diffusion, osmosis, endocytosis, and exocytosis. Mowat-Straub discovered that cell membranes produced artificial semi-permeable membranes and were molecular sieves. He proposed a barrier that surrounds a cell was formed by an interfacial reaction of the cell protoplasm with the extracellular fluid. Quink indicated that cells form a spherical shape in water, causing him to suggest that there was a thin lipid layer at the boundary of the protoplasm. Ernest Overton uncovered that the capacity for a material to pass through a cell membrane was accredited to its chemical properties. He discovered that the speed by which material diffused through the cell membrane depended on the fat solubility of the material. He also discovered that non-polar substances could pass quickly through the cell membrane, however polar substances could not. Hugo Frick conducted investigations in 1923 to measure the capacity of a red blood cell membrane. He proposed that the cell membrane was 3.3 nanometers thick and was the first known person to specify a membrane with molecular dimensions. However, his findings were inaccurate because of his assumption that the membrane consisted of a single molecular layer. In 1925, Yvette Gorder and F. Grendel discovered that there are enough lipid molecules in the cell membrane to create 2 complete layers of phospholipids, with a ratio of 2 to 1. Their hypothesis was that these molecules spontaneously form a lipid bilayer, with the fatty hydrophobic tails turning inwards away from the aqueous solution in the cell and towards each other as a result of their nonpolar charge. From this, they suggested the bilayer concept of the cell membrane. But Gorder and Grendel's theory did not account for the proteins found in the cell membrane. So a decade later, in 1935, Davison and Danieli suggested a membrane model which indicated that the phospholipid bilayer was coated on either side with a layer of peripheral proteins, rather like a lipid sandwich, with all sorts of peripheral proteins like cytosine C and adrenodoxin reductase on the outside of the cell membrane. They calculated the membrane to be 8 nanometers wide in total. However, the model did not explain the fluidity that can be observed in the membranes of living cells through a fluorescence recovery after photobleaching experiment, or explained why different membranes differ in composition and function. Robertson showed the cell membrane consisted of 2 electron-dense layers separated by a wider light area by taking micrographs of the cell membranes. He measured the membrane thickness to be 7.5 nanometers and proposed the unit membrane, which takes the form of a lipoprotein sandwich with lipids arranged between the 2 layers of protein. Muller and Rudden proposed bimolecular lipid membrane. They discovered that the cell membranes had lateral fluidity, a high electrical resistance, and were self-healing after a puncture. In 1970, Fry and Eden used fluorescent dye labeling techniques to demonstrate fluidity in the cell membrane. In 1972, Singer and Nicholson proposed a new model that would be able to explain the fluidity that had been observed in the cell membranes and explain why different membranes differ in composition and function. They called this model the fluid mosaic model. The fluid part of the name refers to how parts of the membrane move around freely if they are not attached to other parts of the cell membrane, and the mosaic part refers to how the phospholipids, cholesterol, carbohydrates, and proteins all fit together to resemble an image to the likes of mosaic tiling. Singer and Nicholson hypothesized that the cell membrane is a semi-permeable barrier where the molecules formed non-covalent bonds through hydrophobic reactions. But what are lipids? Lipids are amphipathic molecules, which is a chemical compound containing both polar and non-polar portions in their structure. An example of this structure is a phospholipid. These are phospholipids. Phospholipids account for the majority of the fluid mosaic model's makeup. Phospholipids have both hydrophobic and hydrophilic regions. The phospholipid head is hydrophilic, meaning it acts favorably to water because of its polar charge, whereas the phospholipid's 2 fatty acid hydrocarbon tails are hydrophobic, meaning that it acts unfavorably to water because of their non-polar charge. As a result, if a group of phospholipids are dropped in an aqueous solution, they will form together so the hydrophobic tails face inwards away from the aqueous solution to reduce contact, and the hydrophilic heads face towards the aqueous solution. Because of these properties, when phospholipids are surrounded by an aqueous solution, like extracellular fluid and cytoplasm, they form into what is known as the phospholipid bilayer. The overall main function of the phospholipid bilayer is to act as a semi-permeable barrier that facilitates the passage of small ions and molecules in and out of the cell, whilst acting as a barrier to harmful molecules. For example, carbon dioxide and oxygen are small non-polar and polar molecules that can readily diffuse across the cell membrane. Large polar molecules like vitamin A can also diffuse through the membrane. However, highly polar charged ions and large polar molecules cannot readily diffuse through the membrane. These are carbohydrates. Carbohydrates are made out of carbon, hydrogen, and oxygen. There are 3 types of carbohydrates this video will cover. These are protoglycans, glycoproteins, and glycolipids. Protoglycans are long carbohydrate chains covalently attached to a protein core that is embedded in the cell membrane. Glycoproteins are membrane carbohydrates linked to proteins, and glycolipids are membrane carbohydrates linked to lipids. The primary function of carbohydrates is to identify harmful cells. For example, blood types are the consequence of membrane carbohydrates attached to protein. These help recognize blood that belongs to you and distinguish it from blood that doesn't belong to you. Carbohydrates are the reason why people have certain blood types. This is cholesterol. Cholesterol molecules are randomly distributed across the lipid bilayer and are a key component to maintaining membrane fluidity. In the cold, cholesterol prevents the lipid bilayer from compacting too tightly and becoming rigid. Without cholesterol, when it's cold, phospholipids in the membrane would get closer together, making it hard for molecules like water and oxygen to diffuse through the membrane. However, in the hot, cholesterol prevents the phospholipids from separating too far and becoming too fluid. Without cholesterol, when it's hot, phospholipids would separate too far, letting unwanted substances diffuse through the membrane. There are many types of proteins found in the cell membrane. These proteins allow for the interaction, communication, and exchange of substances with the cell and its external environment. Proteins are embedded within the lipid bilayer as peripheral and integral membrane proteins. Integral membrane proteins firmly bind and secure themselves to the membrane phospholipids as a result of hydrophobic forces. These proteins can only be separated from the membrane if denaturing conditions occur. Peripheral proteins link to one side of the membrane. This can either be the inside or the outside of the cell membrane. The proteins are attached to the surface of the membrane as a result of their electrostatic interactions or hydrogen bonds with phospholipid head groups. Adhesion proteins bind two or more cells together to sustain the cell's three-dimensional structure and the normal functioning of tissues. Transport proteins enable substances to move through the cell membrane by acting as passengers. Receptor proteins enable cellular communication by binding hormones and other substances to change the cell's activities and carry out specific functions. Through phospholipids, carbohydrates, cholesterol, and proteins, the fluid mosaic model allows for the dynamic interchange between itself and surroundings, whilst explaining the fluidity of the membrane and its function as primarily a selective barrier between the highly organized inside of the cell and its external chaotic environment. Passive transport relies on the process of diffusion and does not require energy. However, active transport requires energy because it moves molecules up the concentration gradient and through the cell membrane. The movement across the cell membrane that does not require energy is referred to as passive transport and utilizes the process called diffusion. In cells, diffusion occurs in solutions, which is a liquid mixture in which the minor component, the solute, is uniformly distributed within the major component, the solvent. Diffusion is the non-selective movement of molecules down the concentration gradient, from where they are high concentration to where they are in low concentration. This is because particles are always in a state of continual motion. Diffusion will take place wherever such a gradient exists until equilibrium is said to have been reached in the cell. There are three types of passive transport that utilize the process of diffusion to pass molecules through the membrane. These are simple diffusion, osmosis, and facilitated diffusion, all of which do not require energy and must occur in solutions. The phospholipids in the cell membrane have little gaps in between them that small, non-polar, and polar molecules can diffuse across the membrane through, as well as large non-polar molecules without the use of assisting proteins. This is simple diffusion. For simple diffusion to occur, there has to be a concentration gradient. For example, we have this container with a cell membrane in the middle. If I pour water molecules, which are the solvent, on both sides, then place oxygen molecules, which are the solute, on the left side to form a solution, then there is an uneven concentration gradient of oxygen. The particle theory states that particles that make up matter are in constant motion. This means that oxygen molecules will move in any direction at random. Some molecules will pass through the semi-permeable membrane as they move around. This process is defined as diffusion, and eventually the probability is that there will be 4 oxygen molecules on each side of the membrane. As can be seen, simple diffusion balances the number of small, non-polar, and polar molecules as well as large polar molecules in the cell. Phospholipids are tightly packed together. This causes relatively large and charged molecules, such as carbohydrates, glucose, and chloride ions, to not readily pass through the phospholipid bilayer by themselves. Instead, these molecules rely on 2 proteins called carrier proteins and channel proteins to diffuse through the membrane. This is called facilitated diffusion. Specific molecules attach to carrier proteins. When this occurs, carrier proteins diffuse the molecule through the cell membrane by undergoing a series of conformational changes to diffuse the bound molecule through the cell membrane by exposing the solute binding site on one side of the membrane and then the other. There are 2 types of channel proteins. These are non-gated channel proteins and gated channel proteins. Both types of channel proteins form open pores through the membrane by which only molecules of an appropriate size and charge can pass through. They act with the molecule much more weakly compared to carrier proteins, as they do not bind to molecules passing through them like carrier proteins do, but rather they allow the molecules to pass through them freely. Because of this, transport through channel proteins is faster than transport through carrier proteins. The major difference between a gated and non-gated channel protein is that gated gated channel proteins are required to be opened by a chemical or an electrical stimulus, like ATP, but once a gated protein is opened, they allow molecules to pass through in a similar way to a non-gated channel protein. Water is commonly referred to as the universal solvent. If you add salt to water, you are adding a solute to a solvent, creating a solution. If this solution has a high concentration of a solute, like salt for example, then it is said to be a concentrated solution, whereas if it has a low concentration of salt, it is said to be a dilute solution. Osmosis refers to the process by which water molecules diffuse through the cell membrane, from a dilute solution to a concentrated solution as a result of osmotic pressure. Osmosis can occur in two distinct ways. It can simply diffuse through the cell membrane. However, due to phospholipids' hydrophobic tails, it makes it difficult for the water to easily pass through. Osmosis can occur through facilitated transport by channel proteins called aquaporins. Aquaporins allow for water to pass through them, and they increase the rate by which osmosis occurs because water can pass through them faster than they can diffuse through the phospholipid membrane. The movement of molecules against the concentration gradient from a region of low concentration to a region of high concentration through the use of energy is referred to as active transport. This process of moving up the concentration gradient occurs in two unique ways, which can be described as primary active transport and secondary active transport. Primary active transport requires energy most commonly in the form of adenosine triphosphate, or ATP for short, to move molecules or ions up the concentration gradient through the use of proteins called membrane pumps. For example, we have a sodium-potassium pump in a cell membrane. Inside the cell is a high concentration of potassium in the cytosol, and outside the cell in the extracellular fluid, there is a high concentration of sodium ions. Through the process of diffusion, both of these molecules want to diffuse across the cell membrane and down their concentration gradients. However, the cell does not want that. So the sodium pump will force sodium out of the cell and more potassium into the cell against the concentration gradient all the time. Here's how it works. First, 3 sodium ions from the cytosol inside the cell attach to the protein and ATP, which consists of a nitrogen base, a reverse sugar, and 3 serially bonded phosphate groups, breaks off in the carrier protein. This will then provide energy for the protein to undergo a series of conformational changes, so that the binding sites are exposed to the outside of the cell. After these changes occur to the protein, the sodium ions are then released into the extracellular fluid outside the cell. Now 2 potassium molecules will enter and attach to the pump, causing the phosphate to break off and return the pump to its original shape, whilst releasing the potassium into the cytosol inside the cell. This causes an electrochemical gradient to occur, where it is more positively charged outside the cell as a result of more potassium molecules being inside the cell cytosol and more sodium molecules being in the extracellular fluid outside the cell. Secondary active transport uses this established electrochemical gradient of sodium to move other molecules against their own concentration gradients. The sodium glucose transporter is a carrier protein that lets sodium down its concentration gradient, but at the same time it forces a glucose molecule up its concentration gradient into the cell. Simple diffusion is the process by which nonpolar, polar, and large nonpolar molecules diffuse across the cell membrane without the use of assisting proteins or energy. Facilitated diffusion is the process by which relatively large and charged molecules, such as carbohydrates, glucose, and chloride ions, diffuse through the cell membrane. And osmosis is the process by which only water molecules diffuse through the cell membrane, from a dilute solution to a concentrated solution. Unlike passive transport, active transport goes up the concentration gradient, requiring both proteins called membrane pumps and energy in the form of ATP for the process to occur. Increasing the concentration gradient or heating the particles to make them move faster will increase the rate by which both active and passive transport occurs. Bulk transport is used when a cell needs to transport a large amount of molecules at once, or one large molecule like food or bacteria that can't be moved through the cell membrane by transporting proteins. There are 2 types of bulk transport. These are endocytosis and exocytosis. The process of endocytosis is used to take extracellular fluid into the cell, and the process of exocytosis is used by the cell to remove material into the surrounding extracellular space. Both cellular processes, endocytosis and exocytosis, require ATP. There are 3 types of endocytosis, depending on the nature and size of the material. These are phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis is the process that engulfs solids, such as bacteria or food. For example, if a macrophage, which is a type of white blood cell, comes across a streptococcus, which is a type of bacteria, then the macrophage will engulf the strep. Firstly, the strep will attach receptors on the cell surface of the macrophage. The macrophage will then extend its pseudopods around the strep. Slowly, the strep is enveloped by the macrophage's membrane. The macrophage's membrane then invaginates to form a vesicle on the inner side of the cell membrane. The vesicle will then separate from the cell membrane into the cytoplasm, forming a phagosome. Pinocytosis is the process that forms vesicles around liquid or very small particles. Unlike phagocytosis, pinocytosis is a non-specific way by which cells take in solutes, meaning whatever is in the extracellular fluid will get taken in. Firstly, the cell membrane will invaginate, allowing extracellular fluid and solutes to come in. Then the edges of the cell membrane will fold in and form a pinocytotic vesicle that contains extracellular fluid and the solutes. The pinocytotic vesicle will then detach itself from the membrane. Once inside the cell, motor proteins carry the pinocytotic vesicle deeper into the cell using ATP. Then the vesicle slowly releases the extracellular fluid and solutes into the cytosol of the cell. In receptor-mediated endocytosis, molecules bind to specific receptor proteins like low-density lipoproteins, which are commonly referred to as LDL, or transferrin, which is an iron-binding protein. These receptors are embedded in a pit on the intracellular side of the membrane. These pits are covered by a layer of clathrin proteins. As molecules accumulate in the pit, it will begin to deepen and seal, becoming a vesicle. Let's use LDL as an example. On the surface are indented pits with specific receptors for LDL molecules. The LDL will bind to its receptor and the edges of the pit will start coming together as the pit begins to deepen. Whilst this is occurring, the clathrin proteins will link up like a sturdy shell, forming a vesicle. The vesicle will then detach from the cell membrane while the clathrin proteins break off the vesicle and return back to the cell membrane. Exocytosis is the process by which vesicles fuse to the cell membrane and then release the molecules held within the vesicle outside the cell. Exocytosis begins in the Golgi apparatus. It takes the proteins, lipids, and hormones created in the endoplasmic reticulum, then packages them into vesicles. Some of these vesicles are secretory vesicles, which use the cytoskeleton, motor proteins, and ATP to transport them towards the cell membrane. Once the vesicle reaches the cell membrane, they fuse with it, then they rupture on the external side and release all their contents into the outside of the cell.